Hello, everyone, and welcome to this audio podcast. This audio podcast is as part of this series and this series or this edition, the research and reports. This audio podcast is part of a Labostix report, and this audio podcast is part of a ZtormLabostix production. Today we're looking at works from around 2022, works that have been presented, works that have been presented in around 2022 within a pediatric transplantation seminar remit. This overview will look at investigating transplant immunobiology and looking at this as a CKD review. And this overview is a part of a ZtormLabostix Collaborative. So to begin with some contents, there'll be in this overview there will be an introduction, looking at the sort of principles of chemokine action, looking at aims and objectives, looking at transplant immunobiology, and a literature review. Looking at prospective investigations and discussion thereafter, and then conclusions, and then a list of citations or references at the latter part of this overview. So intro, to begin with some intro. So the immune system protects the body from infection by pathogenic organism. It is composed of a complex constellation of cells, organs and tissues arranged in an elaborate and dynamic communications sort of network. The immune system is, in its simplest form, a sort of cascade of detection and adaptation, culminating in a system that is remarkably effective. Leukocyte and lymphocyte migration or trafficking from peripheral blood into affected tissues is an essential component of the inflammatory reaction to virtually all forms of injury, and is an important factor in the development of many diseases. Advances in the past few years, if not in the past in the past decade or so, have highlighted the central role of a family of chemotactic cytokines called chemokines in this process. Cellular adhesion molecules, or CAMs, have also been important in line to allograft rejection or transplant rejection, as these molecules allow protein receptor binding. Healthcare also incorporates another basis called the biopsychosocial model. The biopsychosocial model is a technical term or terminology, or terminology for sort of popular concept of the mind and body connection. The basis was initially developed by a psychiatrist, George Engel at the University of Rochester, and first published in around 1977 in an article of Science. The biopsychosocial model draws a distinction between the actual pathological processes that cause a disease and a patient's or recipient's perception of health and the effects on it, called the illness, or referred to as the illness. There are also theories that directly affect the immune system, and there are investigations that show this to be the case, and referred to as as a specialty, as a link, as a sort of an interlink between three main specialties: psychology, neurology, and immunology, and referred to as psychoneuroimmunology or psychoneuroimmunology, and abbreviated as PNI. So now looking at sort of the principles of chemokine action. So chemokines are released by stimulated cells that act locally. Chemokines are produced by intrinsic cells as well as infiltrating cells. Chemokines act through G protein coupled 7 transmembrane receptors and induce calcium influx. Chemokines also bind to proteoglycans and extracellular matrix. Chemokines induce hepatotaxis, which is referred to as cell migration along surface gradients. Chemokines can activate adhesion molecules. Chemokines can induce oxygen burst and the release of granules and proteinases by leukocytes or white T cells. And chemokines can control aspects of immunomodulation, angiogenesis, that is the development of particular cell and specific cell types, and hematopoiesis, the stimulation of particular cells in line to red cells, in line to red cells, and as part of physiological hemostasis and development. So now looking at sort of the aims and objectives of this overview, which will be to look at transplant immunology and neuroendocrinology, and how both the interlink between the interplay between both would forms a basis of this will form the basis of this overview. So now looking at the literature review around this works. So to begin with, Neveu and Lemot in around 1990 highlighted that the immune system and the central nervous system are known to communicate through the endocrine and autonomic nervous system, primarily the autonomic nervous system, and inform that a complete regulatory loop between immune and neuroendocrine system exists. Wayne and team in around 1990 also informed that there is a molecular basis for bidirectional communication between the immune and neuroendocrine system, and the recognition of such stimuli is converted into information in the form of lymphokines, monokines, and hormones, and a message received by neuroendocrine tissues. Nervous system recognition of stimuli can also be converted into chemical signals that can be relayed to immune cells, resulting in physiological changes. Physiological changes, listeners and viewers, but also pathophysiological changes. Then works by Bedofsky and Del Rey in around 1991 inform interactions that imply the existence of afferent messengers derived from the immune system, such as lymphokines and monokines, capable of integrating immune neuroendocrine circuits. At the time of peak of the immune response to innocuous antigens, as well as following inoculation of infective agents, endotoxins, or tumor cells, immunological cells release cytokines which can stimulate the pituitary adrenal axis. The acute effect of these cytokines is mediated by stimulation of the release of corticotropin releasing factor from the hypothalamus. Then works by Grochowitz Grochowitz and team in around 1991 also highlight that conditioned immunosuppression using a taste aversion paradigm, a taste aversion investigation, part of a taste aversion investigation, have been demonstrated in several laboratory environs, and the team report. The investigation had reported that the survival of cardiac allografts in rodent models can be prolonged by immunosuppression using the immunosuppressant CSA or cyclosporin A as a stimulus in taste aversion conditioning. Conditioned animals receive saccharin as a conditioned stimulus paired with an injection of CSA at 6 and 10 days before transplantation. Early investigation had informed that behaviorally conditioned immunosuppression may have important clinical implication as an adjunct to pharmacotherapy amongst transplant immunobiology. Then works by Grotzel and Sridharan in around 1992 informed that integration between several immunological functions in host defense may reveal novel mechanisms of cellular communication and biological adaptation. Then works by Ottaway and colleague in 1992 informed that many of the alterations in immunity resulting from the central nervous system activity can be both explained and investigated in terms of changes in around particularly around lymphocyte migration in response to endocrine signals, neural signals, and through neurotransmitter release and or contact between nerves and the immune system. Then works by Kropotkin in around 1993 had informed that the immune system is active even in a bidirectional feedback loop. And then works by Klein in 1993 informed that cellular and molecular basis for modulation of host resistance is not fully understood, involves altered functioning of both T lymphocytes and cells of the hypothalamic pituitary adrenal axis. Klein also informed that there is a production of cytokines and hormones produced by the immune system and the brain. Then works by Wayne and Blalock in around 1995 inform or highlight that prospective investigation should seek to investigate into the physiological role that cytokines and neuroendocrines have between immunology and neuroendocrinology. Ferenczek and Shrevnova in around 1997 informed that neuroendocrine system influences and or modulate the immune components that link to physical stress. Danzer in 97 informed that there is evidence that different perceptions of stress have different physiological consequences. Different ways of coping with stress result in different consequences on immunity, the nature and outcome of which depend on the type of immune response. Webster in 1997 demonstrated that glucocorticoids and catecholamines favor T helper 2 cells, or Th2 over T helper type 1 cells or Th1 immune cells and mediators by controlling the production of specific regulatory cytokines. Then Ferenczek in 1998 assert that in the immune system, is a sixth sense and may ultimately provide a sort of novel understanding of physiology required for disease and stress involving the immune neuroendocrine communication. Then Klapik in around 2000 informed that though there are questions that remain unanswered, it appears reasonable to include that depression, for example, and possibly stress factors may affect the use of immunosuppression or IST in transplanted recipients. Then works by Hennig and team in 2000. Hennig and team in around 2000 conduct or carry out an investigation on the mechanisms of changes in lymphocyte numbers after stress, and the team identified catecholamine is linked to stress and found it is of major importance surrounding the frequency of mild leukocytosis. Thus, an increase in specific forms of white T cells. Hennig and team in around 2001 also demonstrated that cortisol seems to play an important role in stress-induced redistribution patterns of CD8 positive cytotoxic T cells. This, however, can be explained by different migration processes between cells, for example, different targets of migration and therefore different glucocorticoid influences on target tissues. Gillard in 2001 informed that a crosstalk between the immune and endocrine systems is important to homeostasis, since the interactions can produce various appropriate adaptive responses when homeostasis is threatened. Hubbers in 2003 highlight that in the field of PNI, psychoneuroimmunology, hormones, peptides, and neurotransmitters regulate the immune system. However, understanding the complex interactions between systems is still a challenge for the 21st century, and pathogens, tumors, and allergens are detected with great sensitivity and specificity by the immune system. George Engel's biopsychosocial paradigm is now one of the sort of main bases or models when dealing with most human diseases and disorders. And the biopsychosocial model encourages reading into areas not traditionally associated to transplant immunobiology. Works from Blalock again in around 2007 also inform that as a sixth sense, the immune system is a means to signal and mobilize the body to respond to these types of challenges. Investigators should acknowledge that the research surrounding transplantation should not be restricted to single organs, and that the psychoneuroimmunology may have a role in prospective investigation, and this may again produce a better understanding of the immune system, and it may be a means of attaining T cell tolerance post-transplant. So now looking at an overview in regard to prospective investigation. So in vitro studies using interleukin 15, interleukin 2, interleukin 15 receptor alpha or receptor alpha, stress cellular adhesion molecules, and pertinent monoclonal antibodies. So for example, anti-CD25 could be investigated with chemokine receptors such as CCR5, CCR7, and CXCR3 in a T cell culture system to understand if this affects T cell migration post-transplant, post-transplantation, and using peripheral blood mononuclear cells or PBMCs will also be advantageous. So, as some discussion now. So, whilst there have been some collaborative approaches between specialties or disciplines and practices which incorporate laboratory investigations, understanding CKD or chronic kidney disease in context of transplant immunobiology is still important, and stress mechanisms aligned to psychoneuroimmunology or PNI are topics again to contemplate across solid organ transplant specialties. Some conclusions. So using chemotaxis assays with flow cytometry would aid to give graphical data and linking specific endocrines such as cortisol, adrenocorticotropic hormone or ACTH, and glucocorticoid hormone with the above could be advantageous. Stress also may have specific effects regarding T cell migration post-transplant or post-transplantation, and so implementing migration investigations or trafficking investigations where pathophysiology and immunology is and are concerned is one of the is and are some of the most useful ways to look at correlations. So this slide, listeners and viewers, is a list of citations of references which can be used to identify literature in around this overview, and also looking at sort of identifying literature sort of more generally around for example, looking at sort of aspects around sort of the aspects around clinical science, and also in regards to the sort of concepts and aspects that have been again highlighted in this overview. Draws this audio podcast overview to a close. This audio podcast is part of a research and report. This audio podcast as part of a Labostix report. This audio podcast as part of a ZtormLabostix production.